Hello everyone, welcome to Life Science with Veena. Today we are going to see one another topic from instrumentation, that is colorimeter. Okay, so we're going to start with colorimeter, and in this colorimeter topic, first we are going to talk about the principle of colorimeter, the principle on which the colorimeter works. We're going to talk about the Beer-Lambert's law, the concept of complementary colors, mechanism of working, and the applications of colorimeter. Okay, so if I talk about a colorimeter, it is an analytical instrument which measures the concentration of colored substances. So please remember, we cannot measure the concentration of colorless substance with colorimeter. We can only measure the concentration of colored substances in a solution, and we can quantify that how much light that particular solution is absorbing. Now, Beer's law is the law which states that, which connects the absorbance with the concentrations. Okay, so Beer's law was given by the scientist Beer, that is why it is called as Beer's law. And it talks about the concentration and the absorb absorbed light by the colored solution. Okay, colorimetry, it is a technique used to determine the concentration of a colored solution, and it measures the amount of light which is absorbed or the amount of light which is transmitted. Okay, now this particular apparatus has a wide application in the field of chemistry, biochemistry, and biology laboratories. So physical chemistry particularly, or analytical chemistry, so it has wide application. So it is very essential to study this apparatus. Now here you are able to see that here is a apparatus, that is colorimeter, which is there, and this apparatus has a digital display, auto zero knob, and there is a slot for the insertion of cuvette, and the you can say the knob for adjusting the wavelengths of particular wavelength. Okay, light of particular wavelength. Okay, so this is how, and these are the cuvettes. Okay, so this is a typical structure of a colorimeter. Here also you can see absorbance readout is there, that is digital display. Wavelength readout is there. You can maximize or minimize the wavelengths from here. You can select the mode, and sample holder, that is a slot for putting your cuvette, and transmission control. This is a typical structure. And if you talk about when the light is falling on any object, three things can happen: either it will be absorbed, or it may get reflected, or it will be transmitted. Okay, so these three things can happen when the light is falling on the object, and based on these three things the colorimeter is based. Okay, colorimeter works. Okay, when the light passes through the colored solution, a part of light is absorbed and the remaining light will be transmitted. So Beer's law and Lambert's law talk about the amount of light which is absorbed and the light which is emitted. Okay, so here you can see that there is a inside the colorimeter, this is how the structural setup is. There is a light source which could be a tungsten lamp or a LED lamp. A light source is there. There is a slit through which the light will pass. There is a lens which will focus the light, and the filter which will select the light of a particular wavelength. Only a monochromatic light will be passed through this filter, a light of particular single wavelength. Here is a cuvette into which the sample is placed, colored solution is placed. Here is a photocell or a galvanometer which will detect the amount of light which is absorbed by the color solution. And here is the output which will be displayed on the digital display. That is your optical density or the absorbance. Okay, so these are the typical components of colorimeter, or the parts of colorimeter. So first, a light source will emit the light of visible range, The slit through which it will pass, the lens will focus that light, and through the filter the monochromatic light will be selected. It will fall on the cuvette. The photocell is going to read that, the absorbed light and the transmitted light, and the output will be given. So this is how the light, the mechanism of colorimeter, or you can say these are the components by which the colorimeter works. So I have already mentioned, that is light source, tungsten or LED lamp, filter, monochromator, sample cuvette, photodetector, and a display or a meter. Okay. Next is, so I'm not going to read everything, but let us check the summary. That is, colorimeter is the instrument which determines the extent of absorption of light. By whom? By colored solution. So we are measuring the absorbance of colored solution only by using a colorimeter. The measurement of light intensity, color, and the concentration of colored solution can be determined. Okay, so when the white light is passes through the filter of given wavelength, so that is used in the colorimetry, and the complementary filter is used. Okay, complementary filter. What is complementary color? The color which is absorbed by the solution is different than the color of the original color of the solution. So the color of the solution is different, that is the visible color to your eye, and the color which it is absorbing, that is completely different. So that is complementary color, and complementary filter is. So for example, here if the color of the solution is blue, the complementary filter yellow will be selected, or red will be selected. Green, the complementary is red. So this is how the complementary colors are. So light when falls on the solution in the cuvette, the some light is absorbed depending on the concentration of the analyte, and falls on the photocell, which generates an electric current and will give the optical density and absorbance, that is absorbance. Okay, so this is how it works. Now if you talk about an electromagnetic spectrum, so it ranges from microwaves, radio waves, microwaves, then infrared, then comes visible range. So colorimeter works only in the range of visible light, that is only a small portion of electromagnetic spectrum, that is Vibgyor, okay, which are the colors of the rainbow from right from 400 nanometer to 700 nanometer. So you will be able to see the wavelengths from 400 nanometer to 700 nanometer in the colorimeter. So colorimetry is a use of human eye to determine the concentration of colored species, and spectrophotometry is for colorless solutions. So here is a visible spectrum right from violet, indigo, blue, green, yellow, and orange and red. Okay, Vibgyor. That is the visible range right from 400 nanometer to 700 nanometer is a visible range in which the colorimeter works. Okay, so two laws are there which are given by the Beer and the Lambert. The first one is when that is given by the Beer. So what he said, when a monochromatic light passes through a colored solution, a light of a single wavelength, a single colored light passes through a colored solution, some of the specific wavelengths of light is absorbed, and that is related to the color intensity, that is related to the color intensity. Okay, so here you can see I0, that is the light which is incident, which is falling on the solution in the cuvette. This is yellow colored is cuvette and the solution of concentration C. C stands for concentration, and I is the amount of light which is transmitted, and B is the path length, that is the length of the cuvette or the diameter of the cuvette. So I0 is the incident light, I is the transmitted light, C is the concentration, and B is the path length. Now, based on these factors, the law, Beer's law and Lambert's law was given. So now what does the Beer's law says? So Beer's law says that when a monochromatic light passes through a colored solution, the amount of light transmitted decreases exponentially with increase in the absorbance of increase in the concentration of colored substance. So the Beer's law states that you can graphically represent this as when the concentration of substance is increasing, concentration of colored solution is increasing, the OD or the optical density or the absorbance of a substance also increases. So that is stated by Beer. Okay. Beer is also stating that when the concentration of substance is increasing, the amount of transmitted light decreases exponentially like this. So you will get a curved graph. So that is the statement given by the Beer. That is, in another way, you can say that the amount of light absorbed by a color solution is directly proportional to the concentration of substance in the colored solution, right? And you can also say that the concentration of a substance or the absorbance of a substance, absorbance of a substance is inversely proportional to the transmittance. That can be this interpretation can be drawn from the Beer's law. Now the concentration of a substance is directly proportional to the amount of light absorbed, or it is inversely proportional to the logarithm of transmitted light, because it is inversely proportional and exponential decrease is there, so it is logarithmically inverse relationship of absorbance and the transmittance. Okay, so here A is directly proportional to C, but A is logarithmically inversely proportional to T. Now here you can see the graphical representation: concentration in mg per deciliter versus absorbance, that is optical density. You will get a straight line graph in the Beer's law. Whereas for transmittance versus concentration, you will get a curve. Okay, this is a curve, decreasing curve. Now let us move on to the Lambert's law. Now what does the Lambert is saying? Lambert is talking about a path length. Lambert has also said the same thing but in a different way. When a ray of monochromatic light, so that line is constant. When a ray of monochromatic light passes through a colored solution, or here the word has been used, absorbing medium, its intensity decreases exponentially, as we have seen. The transmittance, that is the intensity of light, decreases exponentially as the length of the light path through the light absorbing material increases. As the path length is increasing, that is this particular thing, that is the path length, as the path length is increasing, the absorbance is also increasing, the transmittance is decreasing. So it can be stated in that manner. So absorbance is directly proportional to the path length. That was stated by the Lambert. Now here also you can see Lambert has said that the amount of light transmitted decreases, transmission decreases exponentially with increase in what? Increase in path length or the diameter of the cuvette or the thickness of the colored solution, concentration of the colored solution. The same thing has been said in two different ways. Beer has talked about absorbance and concentration and the transmittance, whereas Lambert has talked about a path length also. He has considered path length as one of the factor for the absorbance. Now, if we combine these two laws, we'll be able to see that as Beer-Lambert's law is thus expressed as amount of light transmitted through a colored solution decreases exponentially with increase in the concentration of colored solution and increase in the concentration of colored solution and increase in the path length of cuvette or thickness of colored solution. So what the law is combiningly stating that first one, the amount of light transmitted through the color solution, so transmittance is decreasing, absorbance is increasing with increase in the concentration, right? With increase in the concentration. So concentration is also increasing, absorbance is also increasing, but the transmittance is decreasing and the light intensity is also decreasing. So these two parameters are increasing and these two parameters are decreasing. That is, light and transmittance are decreasing and concentration and absorbance are increasing with the increase in the path length. Okay, this is how the combinedly this law can be represented. Now the same thing once we have seen in the sentence format, once we have seen in the graphical format, and now we're going to see it in the equation format. So here you can see the equation, that is Beer-Lambert's law can be represented as A is equal to ABC, where A is the absorbance, capital A is equal to absorbance, small a is the molar absorptivity of that particular solution. So how much light 1 mole of a substance is absorbing, that is called as molar absorptivity of that particular solution. So that will differ from solution to solution, which can be expressed in moles per liter per cm cube, and it is also called as molar extension coefficient. It can be also denoted by the symbol epsilon. Okay, it differs upon the material being studied, that is the solution being studied. B is the path length. Okay, path length is the length of the cuvette or the diameter of the cuvette. This is the path length represented by letter L, and here B, in this equation it is B. So with different cuvettes it will be different. C is the concentration of the sample, that is mole per liter. C is the concentration of sample, that is mole per liter. So this is how the equation can be represented as absorbance is equal to, it depends on three factors. First one is molar absorptivity, second is path length, and third is concentration of a colored solution. Or it can be represented by epsilon, that is molar absorptivity of a solution, concentration, and L stands for the path length or the length of the cuvette. So this is how the absorbance can be determined using a colored solution. Okay, now we will see some questions related to Beer-Lambert's law in the next session. Meanwhile, stay tuned and watch Life Sciences with Veena for more such videos. Thanks a lot. Please like, share and subscribe to my channel. Bye bye.